10 after 9 p.m., depending on where you are. So I'd like to welcome everyone to MVZ Lunch. I don't have much in the way of announcements. I just wanted to remind everyone that although the city of Berkeley and county and state lifted the indoor mask mandate today, campus still has an indoor mask mandate, and so you should still be wearing your face coverings indoors. My understanding is that that is expected to be lifted at the beginning of March, but for now we're still continuing as we were. Let me just ask if anybody else has any announcements. Okay, if not, I will turn it over to Frederick Thunesen, who's going to introduce today's speaker.
Great, thank you, Michael. So it's a great pleasure for me to introduce Professor Dr. Manfred Gahr, who's also a good friend and collaborator. And we were just lamenting before the official introduction, so I'll continue to do this. It's really too bad that Manfred is not able to visit us, but he's promised that he's going to come back, and so we'll be welcoming him hopefully soon and see him in person. I think we're all suffering from some form of long COVID with different symptoms, and this is kind of one of them, so we'll have to catch up at some point. So currently, Manfred is the director of the Max Planck Institute of Ornithology, which, as you can see in his slide here, is now growing and changing its name to Biological Intelligence and joining with the Max Planck of neurobiology. He is also the head of a large lab in behavior neurobiology. I'm going to do this kind of 6 degrees of separation and to tell him that he's also one of ours. So Manfred did his PhD at the University of Kaiserslautern and Caltech with Mark Konishi. And you guys know that, or might know that Mark Konishi was a graduate student here at UC Berkeley with Peter Marler a long time ago. So in that way, there are 6 degrees of separation here. Manfred is actually one of ours. After his PhD, he did a postdocs at Cambridge University, University of Texas, and Harvard before going back to Germany and becoming the director of the Max Planck at Seewiesen. He has 198 articles. If you look at his as a Google Scholar and look at his work, it's extremely impressive. I think it's also kind of reflects a little bit the spirit of integrative biology in the sense that he's been studying the effect of hormones in behaviors and neurocircuits and physiology using a very integrative approach, using many species, mostly in birds, so in songbirds, but also in subalcynes, which are the family of songbirds that are not vocal learners, and in hummingbirds and in Japanese quails, and I'm sure in other animals that I'm forgetting. He's used a range of techniques, from genetic techniques to anatomy, physiology, neurophysiology, behavior work, both in the lab and in the field. And without saying much more than that, I'm going to turn it over to Manfred, and the title of his talk, as you can see, is Hormone Dependence of Birdsong. Thank you, Manfred, for coming and giving a talk to us.
Yeah, thank you again, Frederick, for the invitation and also for the kind introduction, and obviously your effort to link me to Berkeley. So I indeed hope to visit later this year, and I'm looking forward to see your great campus and your great institute. So to start, I choose these 2 photos of our institute just to make the point that we live in changing environments, and since birdsong is sort of a form of sexual social signaling, environment matters very much, at least in our area here in Central Europe. Most birds start breeding around now and end in early summer, so they are photoperiod dependent. and starts when long day shifts to short day shifts to long days. And so the hormones are a way to link the environment to the reproductive physiology of these animals. So I first show you a few examples of hormone dependency of vocal pattern in different species in the northern and southern hemisphere and in the tropics, and then like to come a little bit to mechanisms discussing the role of hormone receptors and some genomic adaptation for the control of song behaviors, and then show you some or introduce a few experiments that we started in the last years to understand really how hormonal activity in the end modifies the behavior readout. So one of our birds is this canaries. So the canary is a typical seasonal bird depending on the photo period. So they start breeding about mid of February, March or so, when days become longer slowly. So essentially what you see here, I hope you can see the cursor. So what you see here is a song of a breeding period. So that's a so-called sonograms, a time frequency display of the sound. And essentially, in the breeding period, the song is different from the so-called non-breeding period, and that can be measured in different ways, like the repertoire here on the top, which is larger in the breeding season, and the song duration is longer, and the amount of so-called non-repeated syllables is shorter. And all this correlates with a large seasonal change here in the testicle size. This is a testicle of about April, and this is one, the lower one is the testicle of September. And these different sized testicles produce different amounts of testosterone, which is shown here in the lower right side panel. So the idea is that seasonal change in testosterone production triggered by the environment modifies somehow the neural circuits that control singing in a seasonal way. So not only the song pattern might change, but also a specific vocal pattern might emerge on the long day high testosterone levels. Like here's the so-called sexy syllables, which is a sort of specific sounds that female canaries like, which has been shown first by Kreuzer and Ballet in Paris, and which we found that they are more abundant in breeding season as compared to, let's say, September, October, at least in the northern hemisphere. Then another example now from the southern hemisphere, just to make the point that this is not a specific feature of northern hemisphere endemic species. So this is a white-browed sparrow weaver that we study in South Africa. So this animal lives in small colonies and has a nice feature that they have two song types. One is the so-called duetting song, which is performed by all members of a group. And then there's a second song, the so-called solo song here on the top, which I just may play so that you get an idea. Okay, so that is a solo song, and it goes on for maybe half an hour or so, so we don't listen throughout it. But it's produced by the dominant male in the breeding season, which we measured here in this or in this diagram. What you see is a presence of solo songs in blue, in red, and absence in blue. And you see that in certain months of the year, from let's say spring to late summer, they don't produce solo song. So, and we can, in relation, we can also trigger this song type by testosterone treatment. Okay, so it's not only a thing of the northern hemisphere, and that also occurs in the southern hemisphere, and seasonality obviously also happens in the tropics. Only now the day length is less important as a zeitgeber, but the rainfall might be the zeitgeber. This is our research place near the Amazon in northern part of Brazil, here in the dry season, and then the same place in the rainy season. And you see there's quite some difference, and the species that we study there is a so-called silverback tanager, Traupidae, which you see here up. And these birds have a so-called dawn song, which is only produced in some parts of the year. Here in this, from essentially July to January, and this is linked to reduced rainfall. If rainfall goes down, dawn song starts, and if rainfall goes up, it ends, sort of. And this is linked to the period when the testicles increase and when testosterone levels go up. So they go up when the rainfall goes down and start going down when rainfall goes up. And so again, the seasonality also, this is very close to the equator. So photoperiod might not play a very big role. Also, there is like a few minutes difference in day lengths. Okay, and then more conceptually, what we see if we look on seasonal singing, in at least in songbirds, and look into males and females, this is sort of the result of a literature review which I did some time ago. We see in different patterns, like in males, we see bird species that have a one period of the year, like the red line, where most singing occurs. One example is the creatid in Europe, and then outside of the breeding season, they don't sing anymore. Then other species, like the black line, the European robin, has two singing periods, one in the spring and another one in fall. And then there are other species like the house wren, which sings more or less, although year round. Also, some highest song activity occurs in the one season, which is coincides with the breeding season. In the female, there are several. You might know that in actually many species, the females are singing too. Only there are additional patterns. There are some females which never sing, like the zebra finch, well known from experimental studies. But others, like the dunnock, has the same seasonality, where there's one peak of singing, and other species, like the female European robin, actually only sings. So this is this purple line here, only sings outside of the breeding season, never in the breeding season. So the only point I want to make, there are different forms of seasonality concerning the singing activity of male and female birds. So if we have always to be a little careful to generalize things, since different species evolve different strategy of seasonality, and this might have consequences for the underlying mechanisms, obviously. Okay, then the same holds then if we look what is really hormone sensitive. And essentially, if I talk about hormones, most cases, I mean, as said already, gonadal steroid hormones, testosterone, and its estrogenic derivatives. So here is again a literature sort of summary about different species that has been studied over the years, songbirds and also non-songbirds here on the lower end of this table, and then different vocal parameters from song rate to frequency range. And then you have this X's. What you see is that some features have been demonstrated to be sensitive to hormone, and others have not been demonstrated to be sensitive to hormone. And in many cases, only some factors have been studied. But what is nevertheless true in most species is that song rate, so the amount of singing, depends on testosterone. So that is testosterone is a red thing. So some data in the zebrafin shows that it's sensitive to testosterone and estradiol, so it's yellow. And then there are some data that song features might depend on estrogens, which is, for example, the case in the canary, but I will come to this again later. But nevertheless, what is certainly correct to say is that the sensitivity of vocal pattern is species specific. So in certain species, the syllable rate might be testosterone sensitive, and in other species, the song duration might be testosterone sensitive, and so on. Okay, so the next sort of introductory point I like, or conceptual point, which is obviously important for the complexity of the mechanistic studies, is the way how these hormones act, and the way is slow. So one sort of typical models that we study are these canaries, and frequently we use female canaries because they have the advantage that they don't sing in most cases. So we have sort of a defined baseline, though. But if so, this bird makes sort of brief calls which sound like this. So very short sounds. So, but if we start treating them now with testosterone, then after about 2 weeks or 1 week to 2 weeks, they develop this type of songs here in the middle depicted sonogram. And then if another 2 weeks later, then after about 1 month, plus minus, then these birds now sing these very organized songs, which sounds like this. Okay, so essentially, what happens is that the testosterone, to really unfold the whole behavior, needs many days to weeks. And this already tells you that many different things might happen in the central nervous system of these animals to organize the underlying song control circuit in a way that this very sort of structured behavior comes out. A stepwise process that always goes first through this levels of less organized vocal readouts. And that means that to understand this whole stuff, we have to sort of study mechanisms over a very long period in the end to maybe get an idea what's going on. Well, then I want also to mention another hormone that might not necessarily be the first one to think if we are talking about seasonality, but nevertheless might be important even for seasonal events. This is a different hormone type which has to do with circadian activity, which is melatonin. And just show a few data on this, just to also tell you that the hormone control of the song might be complex and might depend on integrative activity of different hormonal systems. So in the many years or now ago, again, we found that melatonin is relevant for the song pattern. So here what is shown is in the upper part, the song of a male 1 day before we manipulate the melatonin production. And we do this with a pharmaca, so melatonin is produced in the night, so it disturbs this production or the receptor activity. And then after this nightly treatment, then the song which you see in the middle part of the table is very short, so the animals cannot repeat anymore the segments. And then in the lower part you see a song 1 day after this, or the song after a normal night is again looks perfectly normal. So this indicates that melatonin somehow on a daily basis affects the vocal pattern, next to the testosterone that does it on a very slow, in a very slow manner. Okay, so sort of conceptually, this means we have on one hand seasonal plasticity, singing disappears or certain song types disappear or song pattern might change seasonally, which is a very slow on-off process and which depends largely on testosterone and estradiol. But in integrated into this are also daily changes that are likely due to melatonin, which are much faster, much fast on, fast off, but nevertheless are sort of intercalated with this more slower seasonal effects. And I just show you a little bit something on melatonin first, and then we go to the testosterone and the seasonality. Okay, so mechanisms. Again, so mainly we want to understand mechanism. And so for the melatonin, now here you see above the melatonin, and here we have a schematic view of a songbird brain, and all these sort of areas are part of the so-called song control systems that has been described by the Nottebohm lab and others later in the last 30, 40 years. And now indicated here are two areas, this one and this one. This is a so-called HVC. This is a RA. Oops. And they express now melatonin receptors, the so-called melatonin receptor 1B. And well, so certain cells, we know exactly which cells have the receptor, and now we like to see what, and I showed you already, it has an impact on the song patterning, but one way to at least show that the melatonin might do something on the circuit directly is to study these neurons on the auditory vocal in the paradigms that is shown here. So what we do here is we study, I think these are RA neurons. This is work by a doctoral student, Susie Seltman, by monitoring HVC activity while the bird is behaving. And we do this with the small helmets, which you see here on the head. So the bird has an electrode implanted in the brain, and then we use some telemetric device to transport the brain activity to a receiver. And so just to show you this data here, you see the activity at a certain neurons that we record, so these are extracellular recordings during singing, and then here in daytime, and then you see here the same neuron when we play back the song. So that is a particular way to test HVC neurons. You can play back the bird's song to the bird and then ask the neuron if they respond or not. So, and clearly in the daytime, these neurons that has been known don't respond. Now, if we do the same during the night, then the neuron nicely respond to the playback. So this vertical line is on zero is onset of playback. So if the neuron certainly respond to the playback. But if we now during the date, so it could be night or melatonin, and we thought it's melatonin, could also be other stuff, obviously. But so if we now add in the daytime melatonin and do the playback, then the result is essentially like in the night now, so this HVC neuron, RA neurons respond to the playback. And in the lower parts, this is only sort of many repeats of this trial, like 240 repeats. And you see here, this is playback onset. There's a clear difference as compared to the situation where we have no melatonin. Now, this does not tell us really how the melatonin affects these neurons for the singing, but as said, the same neuron is involved in singing and in auditory playback, but we don't really understand what the melatonin does to these neurons in the night to affect the song pattern, but we know that the melatonin affects the circuit at least. So it's at least goes into the right direction, so to speak. Now let's move then to the seasonal plasticity, and now for the rest of the time I'm talking on testosterone and estradiol as hormones. And just again some background information for you. So testosterone can act on the brain via the so-called androgen receptor, either as testosterone directly or after metabolization into dihydrotestosterone. And the androgen receptor effect is a transcription factor, intracellular transcription factor that affects gene expression of particular genes, those that have a so-called androgen response element. That's a particular sequence in the promoter. So this testosterone binds to the androgen receptor. You can affect certain types of genes. Alternatively, testosterone can be transferred into estradiol or estrogens in the brain or in other organs, and then activates the so-called estrogen receptor, again a transcription factor intracellular that binds to the DNA, but now recognize other sequences in the promoter. So testosterone via estrogen receptor activity will activate different genes like these and via this pathway. So that means the hormone via the particular receptor can activate certain genes in a differential way. So much about the background. Okay, and then obviously in the song system, we have not only the melatonin receptors, which I showed you before, but now we have this androgen and estrogen receptors. So here again, these areas are all song controlling areas, and the blue dots represent androgen receptors. So this is an old in situ from the times when we still did a radioactive labeling. So this is HVC, this area, and many androgen receptor expressing cells, but as all other areas of the song system also have these androgen receptors. But in HVC, in addition, you can also find estrogen receptors, which is sort of is the blue cell, so it's the red cells in a somewhat lower density. They occur here. So essentially, we have one part of the circuit that has estrogen and androgen receptors, and all other parts have androgen receptors. Okay, now I first want to talk a little bit for 5 minutes or so on let's say the receptor itself and how. Oh yeah, no. First, I like to show you a nice little film. Now, making a small excursion to the chicken to show that the receptors are important, and play you the film. And so just listen, and then you see the chicken crowing, and okay. Okay, so you heard a lot of background, zebra finches, and also and what have you in this aviary room, and so you saw the chicken crowing but no sound. And the reason for this is that we mutated, made the androgen receptor function less, so that the animal that was a result can still do the movement, but this bird did not produce sound. So just demonstrating that, for example, in this bird, the sound production depends clearly on the androgen receptor. Just to make the point that these receptors are really crucial for sound production. Okay. Then another point I like to make is, I said in the beginning that the effects of the hormones are very diverse between species. Now, well, and this comes in coincides with the observations that the receptor distribution differ between species. So, and that concerns mainly the estrogen receptor. Now, what I is showing here is a cartoon. This is represents a canary, HVC. This is a zebra finch. Here, these are the blue cells. Again, the androgen receptor of a canary. Now, this is essentially very similar in most species that we looked on, so canary and the zebra finch. But then, if we look on the red symbols, these are cis cells here. This is the estrogen receptor, in this case of a gray tit, just to show you some diversity. Oops. But this receptor is expressed in a more species-specific way. So for example, in the zebra finch, there are very few such receptor in HVC. In the canary, there are many such red or estrogen receptors in HVC. In the gray tit, sort of is in between. So we have differences in the receptor sensitivity of these HVC neurons, and maybe also concerns other parts of the system. But that's so if you have no receptors or many receptors or few receptors or in different cell types, the effect might be different. Okay, so then the other thing that matters is the genomic sensitivity of genes to the receptor. I told you that this androgen and estrogen receptor are transcription factors. So that means they have to bind to certain parts in the promoter, so the regulatory parts of the genes. And what we see then is that this differs between species. For example, what is shown here are some genes that we found in HVCs, and then we look into the gene of a canary and the zebrafinch in this case, just for comparison. No? And then, for example, we find such a estrogen response element in this symbols here, the promoter region. Or another scenario is we find additional promoters that are not around in the zebrafinch, but are only around in the canary. So that means if we have a particular gene, then it could be, in this case, estrogen sensitive in the canary, but not in the zebrafinch. Obviously, it could also be the other way around. I don't want to say that the canary is now really more hormone sensitive than the zebrafinch. I want to make the point that you can, certain genes or gene networks might be hormone or testosterone or estrogen in one species, but not in another one, which leads to species differences in the effects that these hormones might have on in this case, HVC or RA. By the way, here, this is such a HVC. I forgot now which of the genes it is actually. Okay. Now the rest of the time, the last sort of 10 minutes or so, I like to spend now on the canary and sort of try different approaches that we try to combine to understand really how this testosterone activity, either as a testosterone or as an estradiol, unfolds the neural mechanisms that in the end lead to the singing. And again, what we try to do is we have here that just represent songs. I will explain it in a second. We try to understand what the gene networks are doing, or which gene networks are regulated by the hormones, which then impacts, for example, neuron morphology or neuron physiology, and then try to predict something for the behavior. And as I said already in the beginning, we don't really understand this, but we try sort of to home in. And the complexity is that you have many cells or many neurons, and things happen over a very long time until the behavior can be fully displayed. Okay, again, our canaries, just to remind you about the problem: big testicles, small testicles, big testicles, much hormone, testosterone, little hormone, much testosterone, very structured songs, little hormone, not much structured songs. Okay, so one thing is we can in the canary nicely distinguish androgenic and estrogenic controls of a certain vocal pattern. I told you this already, that the repetition rate depends on estradiol. So that's also some older work of my former student Leonida Fusani. So here you have a song induced in the female by testosterone, and then if we block the conversion of testosterone into estradiol with a certain substance, it doesn't matter what it really means, then the song pattern is changing, and these animals are unable to repeat so-called syllables here with very high speed, and not for a very long time. So it's more sort of less structured than in this situation. That can be measured. So these repetitions are called tours. So the tours with more than 10 syllables per second repeated, so more with 10 hertz or so, are less common if we block the conversion of testosterone into estradiol. So that means if we ruin, so to speak, the estrogenic pathway, then the birds are still able to sing, as you can see here, and the song is also okay, but they are not able to produce certain features, such as this very fast repetitions for a long time, which is an estrogenic pattern. And this is relevant since if we can sort of home in on estrogenic vocal patterns, this makes our life a little easier, since I told you that we have only one part of the entire circuit that makes this estrogen receptors. Okay, now to come to the genes, we do the following. We sort of isolate HVCs and do this at different times after testosterone application, 1 hour, 3 hours, 8 hours, 7 days, 14 days, 30 days, and so on. And then study the transcriptomes of these neurons. And I don't want to go in any details. I just want to say we see very many genes that are regulated. Some are regulated constantly up, others are constantly down. Some are going up and down, and very many are regulated only at certain time points, like 3 hours after testosterone or 7 days after testosterone. So, meaning that there are many candidate genes that might do something to the development of this HVC here, and for example, for these cells at different time points or during the entire time. And so yeah, we actually we have no means currently to study this complexity really, but nevertheless we can sort of single out some genes or some processes and try to see whether those matter. And so that's what we do. Like we try to see genes that are upregulated not only under testosterone treatment, but maybe under testosterone, not only in testosterone treated females with but also in males, or also in castrated males following treatment. So we try to sort of have different experimental groups to reduce a little bit the number of potential genes, and then select at the moment those that are upregulated when the sort of the song is well structured. Now, one such gene is a well-known gene. It's called BDNF. It's a brain-derived neurotrophic factor. And this we like this one in this list. Many others, because we know already since a long time that it's regulated under estrogenic conditions. So here again, that is our HVC now as an in situ hybridization for BDNF. And now if we treat then the birds with this inhibitor of the estrogen pathway, then this gene is not expressed anymore in HVC, which is here. And so that's with testosterone but no estrogen production. Whereas other genes expressed in the same area, so this is a parallel section, like this one here, an ATP synthase, are not affected at all under if we take away the estrogen. So it's an estrogen regulated gene, is BDNF, and we also know that it matters for the singing. So that is what it's a little complicated. So so the BDNF is originating in endothelial cells, which we can sort of suppress in their activity using this substance. So essentially what happens is if we have no testosterone, then the females are not singing. If we get testosterone, then they develop a number of syllables, songs they sing. But if we suppress in this group here the BDNF production, then song is very rare. But if we substitute with BDNF in this group and in this group, particularly in this one where we first block the BDNF production, then we get very nice song repertoires, and also in this part large tours with repeated syllables. So suggesting that, and this BDNF availability is done with some vectors that are expressed artificially in HVC. So means if we bring more BDNF to this HVC, then the birds develop some decent song pattern. And so currently, or not currently, since 2 or 3 years, we try to understand what the BDNF is then really doing, but this is a very difficult project and we don't have a final result yet. But the point is we I have our transcriptomes triggered by estradiol or testosterone, single out genes or groups of genes, and then try to study the impact on the network and the behavior. Now, a different sort of thing is the effect on morphology. So just show you different levels of analysis. So one is more on behavior and the molecular mechanisms. One other one is the effect on the neuroanatomy. Now here this is done like this. So we give testosterone, then the bird starts singing. So here syllable rate, and this is time. So this bird has three different syllable rates, a slow one, a middle, and a very fast one. And then we can end the whole thing here when we remove the testosterone. If we give the testosterone again, then the whole thing starts again. So without testosterone, there's no song. Now, what we did here now is a very detailed analysis. We look now into a spine density. And the reason for this is because our transcriptome analysis told us, okay, testosterone affects neuron morphology, so and neurite and the neurite. So we look, so these small things here are so-called spines, and with testosterone they become fewer. So that's a sort of this group. Then you give testosterone, then fewer spines in a certain cell type. And even if we remove the testosterone, then this just stays. And so that's specific for this HPC. So that means that during the first induction of singing, the circuits with HPC neurons change, and then they just stay like this. And nothing happens anymore later, even if you induce song a second time. And this is just a quantification. So this is a distribution of spine per micrometer of dendrite, and then you see it shifts to the left, so less spines per dendrite, so to speak. Okay, just to tell you that the hormones affect the morphology, and this can be permanent, even if song ends when we take away the testosterone, but the morphology just stays. So, and the last level of analysis that we do is electrophysiology, or using either electrodes or in this case calcium imaging. So in this, what we do here, we look into this HPC, where we label the cells with a certain sort of indicator that indicates activity. It's a calcium sensor, and then we can look into this HPC with a portable 2-photon microscope, with which you can see, for example, the neuron stomata, or here some dendritic parts also. So you can see the morphology under some conditions, but in this case we look into the physiology. And as an example, showing here 3 neurons, A1, 2, 3. They're color coded for purpose of this talk. So this, so first of all, these neurons do something when the bird is calling, at least the red neuron. So whenever the bird, this is the onset of a call, then the neuron becomes active, whereas the blue and the green neuron don't do anything when the bird is calling. Now, what we do is this, unfortunately, under these conditions, the birds, well, to get them to record this HPC, we have to have very dark environments, and birds don't, at least these birds don't like to sing in this environment. So what we do then is we record their songs of a day and play back their own song during dark, when we dim the light. This is sort of a specific way to study this HPC activity of the neuron specificity. And so what we show then here, this is again these 3 neurons, 1, 2, 3, and now we play back the song at this day or at that day and then another day. So actually this preparation has the advantage, we can sort of study the same neurons longitudinally. So what you see then is that at the first day, the green neuron never responds to the playback. In the beginning, the blue and the red neuron respond, and then here the response is somewhat damped, but then after even more time under testosterone, sort of the blue neuron is not responding anymore to the playback, but the red neuron still continues to respond. So just want to say what we do is we study the neurophysiology of these neurons over time to see how and if the changes in auditory vocal properties coincide with some changes in the song patterns that happen over time. But again, we don't really understand this. That's the work of my colleague Shuwen Ma mainly. So that is essentially all I wanted to show you. And then just one glimpse to end is that I think to really do things like study sexual behaviors, we have to move to the field. And even if it's not really so doable now, it will be maybe in 5 to 10 years. And just show you a first approach to this. As I told you, this white-browed sparrow, we were singing different songs in different times of the year. So we studied this in South Africa. This is such a colony of these birds. So they have their nests and defend them. And the dominant male sings a solo song, and he sings also a duet song, and all birds duet. So we try to see what happens in the HVC when the bird is doing solo song, and in time what happens when he is not able to do so. But again, we have no real answer, but just show you, because I like it very much, that we can indeed study at least this song stuff in the field. And so here, this is the environment, this is our laboratory in the car, and here you see a bird with a transmitter on the head. And then it looks like something like this. So this bird has a transmitter on his head here, and another one here in the back. That is an audio transmitter, so we can listen to the sound and study at the same time its brain activity. And we do this parallel in 2 or 3 birds. Here in the background, the duetting partner. And he's completely free, sitting in the tree. Obviously, if you fly away, then is ended. But what you can do with this is, here we record the male HVC and then a female HVC while they are duetting. We color-coded the whole thing. So the blue is the male part of the duet, and the red the female part. And what you see is this HVC is active when the male is doing his male syllables, and the HVC is active, female HVC is active when the female is doing the female thing, but not when the female is listening to the male, or vice versa. So this HVC neurons are sort of coding only their part, but not really also following the coding of the parts of the duetting partner, and so on. I don't want to go into details. I just want to make the point that the goal is that at some point we hope to at least do a lot of work in more natural environments rather than in this with the canaries in small cages. So, and then to summarize what I try to tell you, and this is the people that actually did most of the works that I showed you. I try to tell you that seasonal singing depends on gonadal testosterone and its estrogenic metabolites, and this is somehow more also integrated with melatonin activity affecting the song on a more daily basis. And then try to sort of tell you that testosterone-induced singing correlates with massive changes in gene expression in transcriptomes of, for example, this HVC and likely all other song-controlling brain region. And so that we currently have no real hands on studying experimentally all these changes, but can select certain genes or groups of genes to study their impact on the vocal pattern, and particularly on the level of anatomy and electrophysiology. And then also try to say that some reasons for these species differences, likely in timing, but at least in patterning, is due to the species differences in the absence or presence of receptor types and in differences of the genome to these receptors. And with this, I again here my colleagues, Falk and Meki and Carolina did most of the transcriptomic works, Mariana and Falk's anatomy, Shuwen and Lisa and Susanne the electrophysiology, Phil and Connie the behavior. And with this, I thank you for listening.
Thank you, Manfred. I'm sure we have time for questions, so you could raise your hand or put them in the chat, or I don't know if Michael wants to say anything.
No, people should feel free to unmute themselves or put questions in the chat. Thank you so much. That was a wonderful talk.
Thank you. Thank you. Maybe I stop sharing. Is this okay? Yes. Okay.
Manfred, yeah, I have. Or Leah, go ahead.
Oh, well, I was going to say I have a quick sort of methodological question. I assume that a lot of the work that you showed us today were like recordings from HVC, and I was wondering if specifically the melatonin study that you showed, if those recordings were from HVC.
Yeah, we recorded both RA and HVC. I think the cells which I showed you are RA neurons, but we found a very similar pattern in HVC neurons.
And the transcriptome work, was that done with cells from the HVC also, or from other parts of the cell system too?
All transcriptome works, sorry if I was not precise, were all from HVC.
Okay, thank you.
Judy, I think you're next.
Great talk, Manfred. Thank you.
Thanks.
So I had a question regarding the model of the females that we're treating with testosterone. I've always been wondering, like, females are also vocalizing. They also sing in the Canary, for instance, although rudimentary song. Males do sing far better. But then you have a background here of like a brain that has been formed with its own bath of like estrogen and testosterone. And then you're coming on top of this, and it looks like the female model looks like a male, like we're trying to take the female and make it a male one somewhat. That's the feeling that I have, although we're not taking into account the female background. And I'm wondering to which extent this is true and really representative of what's happening in the male brain, for sure. And whether we could also look into the male to like, make it female maybe, or I don't know. But,
sure. I mean, there are two parts to it. I mean, one thing obviously is this testosterone treatment is always highly pharmacological approaches. I mean, it's useful, but obviously the levels are much higher than those as you would ever find in a male, and even certainly much higher than in the female. So we only use it because we can clearly sort of predict the transition points in the behavior. And if we use females pre-selected for non-singing, then we start with 0. And that in the males is very hard to do because at least in the Canaries and a number of other species, they have always some form of song. Only the song is then changing in the breeding season. So we have not a very good baseline, but obviously this is a quite artificial model, and maybe in the long run we should not do this anymore because we might overflow the system with the testosterone. Yeah.
And could you actually remove the testes from the male birds from birth, for instance? I don't know, and then see, play with the hormones at this stage?
Yes, you can, and you can then do a hormone replacement therapy, so to speak. That's what you would do, which is all possible. But we also did those things. But the the castration also has some problems, because you get some sort of the brain tries to work against it, and then tries to release very high levels of gonadotropins, which then stimulates the adrenal glands to produce steroids. So it's I mean, using the castrated males would be sort of equivalent to using the testosterone females. But again, I think it's also sort of a, in the long run, models that we, at some point we should not use anymore, because it's too artificial. I think for the moment it's still fine, either the females with the testosterone or the castrated males with then hormone replacement. But it's far from natural milieu. Yeah.
And is there any hope of developing little sensors where you could measure the testosterone level over time or something in the birds?
Yeah. Good question. We try to do this. So we build a hormone, a remote hormone sensor, but that is a. We are currently, if everything works well, we can put it on a chicken. So it will immobilize the zebra finch or something. But maybe in 10 years, we are, not me, but some younger people will be at the point to have a hormone sensor with which we could really, in a real time, sense testosterone, and then that would be a completely different ballgame.
Manfred, there's a question in the chat. It says, it's Chuck who's asking you, have you had the opportunity to study closely related phylogenetic pairs to identify details of some species divergence?
No, not really. We didn't do this. So closest that we study different forms of Estradiol finches. From zebra finches and related animals, but they are not really so closely related. We have some African species, and the zebra finches are from Australia. No, we didn't. But it could be a good strategy indeed.
I had a question about estrogen versus androgen receptors. You've showed a lot of differences, kind of, at the level, at all levels, right? At the level of like the behavior, at the level of where they're expressed in HPC, and to what extent, and then at the level of the genes in terms of the beyond the receptor when you get the gene transcription. Have you able to correlate correlate all these differences, like the different in whether it's song rate or in song duration is also correlated where it is in HPC or correlated with genes, or are these different kind of differences make sense in a kind of a story of like, oh, this is what androgen does and this is what estrogen does?
Yeah, well, indeed, if the estrogens are relevant for different factors than androgens. So the estrogens, I think, are relevant for sort of more fine-tuning of the system. So even if you take away the estrogens, you still get a large part of the transcriptomes activated, and the HPC also anatomically develops. But there are differences. Some of the differences might concern the spine forming on some neuron types, but there might be others which we don't know yet. But essentially, what the strategy is a little bit what you are saying. We try to titrate out certain sort of gene networks that are, for example, estrogenic, and then with the hopes that we can link them to certain cell types. I mean, that's a big challenge. Oops. That's a big challenge, because what we would need for this really would be cell type specific transcriptomes, which all the stuff which I showed you is not this one. But So it will get even more complicated, because you can already foresee that there will be different cell types that are differently sensitive to estrogens and others. So not all cells that have an estrogen receptor will behave the same way, and not all cells that have an androgen receptor will behave the same way. So in the end, it will be, I don't know yet how many different cell types there will be, and each of them, depending on the receptor level, might then be affected differently. And so that is certainly a big problem. And the other big problem is to bridge from the, let's say, neuroanatomy to the neurophysiology. So the neuroanatomy is sort of simple to study, and then what you can also study is the neurophysiology. But somehow it has to, these are the same neurons, so it should be sort of linked somehow. And it's very difficult to predict the changes in the neurophysiology from the changes in the neuroanatomy. At least we find it very difficult.
Okay.
Well, thank you again for sharing this super interesting research with us. That was just fantastic. Those of us in the museum don't get the opportunity often enough to think about neurobiology and behavior. So it's wonderful to hear about. And we hope to see you in person sometime soon.
Yeah, I hope too. So this is a nice thing about this evening talk. I can go ahead now and have a beer. Because it's 10 p.m. now, so I'm done with my day.
Well, you've earned it. Thank you.
Thank you, Manfred.
Again, thanks for the invitation, and hope to see you. Bye bye. Bye.